This method can help answer key questions in the field of tumor biology about the micrometastases found in colon cancer patients. The main advantage of this technique is that it allows highly sensitive detection of micrometastases induced by GFP-labeled patient-derived colon cancer organoids in the mice.
Generally, individuals new to this method will struggle because it can be difficult to detect colon tumor organoid-derived micrometastases in distant organs in mice.
Demonstrating the procedure with Yu Okazawa will be Kosuke Mizukoshi and Yu Koyama, both PhD students and surgeons from my group.
To generate human colorectal cancer cell organoids, first add 150 microliters of artificial extracellular matrix per well to a 12-well plate on ice. When all of the wells have been coated, incubate the plate for 30 to 60 minutes in a 37 degrees Celsius and 5% CO2 incubator to solidify the gels. Next, resuspend a patient-derived tumor xenograft cell pellet in fresh colorectal cancer organoid culture medium supplemented with 5% fetal calf serum or FCS to achieve a 3 × 10⁵ cells per milliliter concentration, and seed 1 milliliter of cells onto each artificial extracellular matrix. Then, return the cells to the cell culture incubator. The next morning, carefully transfer the culture supernatants, including the floating cells, into 1.5 milliliter microcentrifuge tubes for their centrifuge.